Hi, this is the Geneticist, and I am your host, Zara. Welcome back to Decoding Dynasty, where we unravel the intricate threads of genetics that have shaped the lives and legacies of royal families throughout history. In this episode, we delve into a condition so intertwined with the annals of aristocracy that it earned the somber moniker, the royal disease, hemophilia. Imagine a world of grand palaces and intricate power struggles, where the health of an heir could determine the fate of an empire. We'll journey back in time to explore the startling prevalence of this inherited bleeding disorder among European royal families, tracing its path from Queen Victoria through generations of kings, queens, and czars. We'll uncover how this royal disease wasn't just a medical concern. It cast a long shadow over social customs, influenced marriage decisions, and most dramatically played a significant role in some of the most pivotal moments in European history, including the very decline of the mighty Romanov dynasty. But this is Decoding Dynasty, and our exploration goes far beyond historical accounts. After painting this vivid picture of the past, we will transition to the intricate world of modern science. We'll delve deep into the clinical symptoms that those affected by hemophilia experience, unravel the fascinating molecular and medical genetics that underpin this disorder, and explore the biochemical pathways that are disrupted. We'll navigate the complex cellular and molecular mechanisms at play, understand the pathogenetics, how this disease develops, and examine the sophisticated screening and diagnostic techniques used today. So prepare to witness the powerful intersection of history and cutting-edge science. Join me as we decode the royal disease, hemophilia, and uncover the genetic truths behind a truly dynastic affliction. And now let's begin our exploration. As we mentioned, the story of hemophilia and European royalty is a compelling intersection of genetics and history, most notably linked to Queen Victoria of the United Kingdom. Born in 1819, Victoria's reign spanned a transformative era, but unbeknownst to her and her contemporaries, she carried a genetic secret that would ripple through the royal houses of Europe for generations. While Victoria herself was not affected by hemophilia, she was a carrier of the mutated gene on one of her X chromosomes. Understanding X-linked recessive inheritance is crucial here. As we now know, females have two X chromosomes. If one carries the mutated gene and the other is normal, the normal gene usually compensates, making her a carrier without symptoms. However, she has a 50% chance of passing on the mutated X chromosome to each of her children. It was through Victoria's descendants that hemophilia manifested in several prominent royal families. Let's trace this transmission. One of her sons, Prince Leopold, Duke of Albany, was affected by hemophilia. He experienced the debilitating symptoms of the disorder throughout his life. Two of Victoria's daughters, Princess Alice and Princess Beatrice, were carriers, just like their mother. Princess Alice married Prince Louis IV of Hesse, and one of their daughters, Alexandra, became Tsarina Alexandra Feodorovna upon her marriage to Tsar Nicholas II of Russia. This link introduced hemophilia into the Russian imperial family. Their only son, Tsarevich Alexei, inherited the mutated gene from his mother and suffered from severe hemophilia B. Princess Beatrice married Prince Henry of Battenberg, and one of their daughters, Victoria Eugenie, married King Alfonso XIII of Spain, thus introducing hemophilia into the Spanish royal lineage. Several of their sons were affected by the disorder. The fact that hemophilia seemed to appear suddenly in Queen Victoria's descendants sparked much speculation at the time. While the exact origin of her mutation remains a subject of historical and genetic inquiry, it's understood that such spontaneous mutations can occur. The consequences of this genetic inheritance were far-reaching, extending beyond the personal suffering of those affected. In Russia, as we touched upon, the illness of Tsarevich Alexei had profound political implications. The chronic and unpredictable nature of Alexei's hemophilia created immense stress within the Romanov family. Their desperate attempts to manage his condition led them to seek the controversial counsel of Grigory Rasputin. Rasputin's influence, gained through his perceived ability to alleviate Alexei's bleeding episodes, further alienated the royal family from the Russian public and the aristocracy, contributing to the growing discontent that ultimately culminated in the 1917 revolution. It's a stark example of how a genetic disorder, seemingly confined to the private lives of a family, could have significant ramifications on the course of history. The vulnerability exposed by Alexei's illness, coupled with the perceived mismanagement and the influence of figures like Rasputin, weakened the monarchy at a time of immense social and political upheaval. Similarly, in Spain, the health of King Alfonso XIII's sons affected succession and stability within the royal family. The public awareness of this royal disease also began to shape perceptions of the monarchy in other European nations, raising questions about lineage and fitness to rule. The dramatic historical saga of hemophilia in European royalty provides a compelling backdrop to understanding the fundamental biological processes at play. What was once a mysterious affliction shaping the destinies of nations is now understood through the lens of modern science. Let's now delve into the intricate molecular and genetic basis of this royal disease. Clinical symptoms of hemophilia. Following our exploration of the historical impact of hemophilia, let's now turn our attention to the clinical realities of this condition. Hemophilia is fundamentally characterized by a deficiency in specific blood clotting factors, leading to an impaired ability to form stable blood clots and control bleeding. The severity of symptoms is directly correlated with the level of the deficient clotting factor activity in the individual's blood. The primary clinical manifestation is prolonged and often excessive bleeding. This can occur spontaneously without any identifiable injury or as a result of trauma, surgery, or dental procedures. The nature and frequency of these bleeding episodes vary widely depending on the severity of the hemophilia. A hallmark of moderate to severe hemophilia is hemarthrosis, bleeding into the joints. The joints most commonly affected are the knees, elbows, and ankles. These bleeds can cause acute pain, swelling, and inflammation. Recurrent hemarthrosis leads to chronic joint damage known as hemophilic arthropathy, characterized by cartilage destruction, synovial thickening, and ultimately impaired joint function, pain, and stiffness, significantly impacting mobility. Bleeding into muscles, known as hematomas, is also common. These can cause pain, swelling, and if large enough, may compress nerves and blood vessels, leading to further complications. More serious bleeding can occur in internal organs, including the gastrointestinal tract and the kidneys. Intracranial hemorrhage, bleeding within the brain, is a rare but life-threatening complication that requires immediate medical intervention. Bleeding in the neck or throat can also obstruct the airway and be life-threatening. Even minor injuries such as superficial cuts or bruises can result in more significant and prolonged bleeding in individuals with hemophilia compared to those with normal clotting function. Similarly, surgical and dental procedures pose a considerable risk of hemorrhage and require careful planning and management, often involving pre-treatment with clotting factor concentrates. The age of onset and the initial presentation of symptoms can vary. Severe hemophilia often manifests in early infancy with spontaneous bleeds or excessive bleeding following minor trauma, such as during circumcision or vaccinations. Milder forms of hemophilia may not be diagnosed until later in childhood or even adulthood, often following a significant injury or surgical procedure. It is crucial to distinguish between the two main types: hemophilia A, caused by a deficiency in coagulation factor VIII, and hemophilia B, or Christmas disease, caused by a deficiency in coagulation factor IX. Clinically, the bleeding symptoms are very similar in both types, and laboratory testing is required to differentiate them by measuring the specific factor levels. Molecular and medical genetics of hemophilia. As we discussed in the historical context, hemophilia A and B are inherited as X-linked recessive disorders. This means the genes responsible for producing the deficient clotting factors are located on the X chromosome. Hemophilia A, the more common form, is caused by mutations in the F8 gene located on the long arm Q of the X chromosome at position XQ28. This gene spans approximately 186 kilobases and contains 26 exons. Mutations in the F8 gene lead to a deficiency in functional coagulation factor VIII. These mutations are highly diverse and can include inversions, particularly a common inversion involving intron 22 or intron 1 of the F8 gene, which accounts for a significant proportion of severe hemophilia A cases. Deletions and insertions. ranging in size from small, affecting a few base pairs, to large, encompassing entire exons or the whole gene. Point mutations, including missense mutations, changing a single amino acid, nonsense mutations, introducing a premature stop codon, and splice site mutations, affecting the processing of pre-mRNA. The type and location of the mutation in the F8 gene often correlate with the severity of hemophilia A. Mutations that result in a complete absence of F3 protein or a severely truncated, non-functional protein typically lead to severe hemophilia A. Mutations that allow for the production of some functional F3 protein result in moderate or mild forms of the disease. Hemophilia B, the royal disease, is caused by mutations in the F9 gene, located on the long arm Q of the X chromosome at position XQ27.1, Q27.2. This gene is smaller than the F8 gene, spanning approximately 34 kilobases and containing 8 exons. Mutations in the F9 gene lead to a deficiency in functional coagulation factor 9. Similar to hemophilia A, the types of mutations in F9 are varied and include deletions and insertions, affecting part or all of the gene, point mutations, including missense, nonsense, and splice site mutations, large rearrangements, although less common than the intron 22 inversion in F8. The specific mutation identified in the Romanov family, as mentioned in your research, was a mutation in the F9 gene that disrupted RNA splicing, leading to a severe form of hemophilia B. The X-linked recessive inheritance pattern dictates how these genes are passed down. Males, XY, who inherit an X chromosome with a mutated F8 or F9 gene, will have hemophilia because they have no corresponding normal allele on the Y chromosome. Females, XX, who inherit one mutated X chromosome are typically carriers. They usually have sufficient clotting factor produced from their normal X chromosome to prevent significant bleeding. However, they have a 50% chance of passing the mutated X chromosome to each of their children. For a female to manifest hemophilia, she generally needs to inherit a mutated gene on both of her X chromosomes, homozygous or compound heterozygous. This is a rare occurrence, more likely in situations of consanguinity, although, as we noted historically, this wasn't the primary driver in the European royal families. Other mechanisms for hemophilia in females include skewed X inactivation or chromosomal abnormalities involving the X chromosome. Carrier females can sometimes have lower than normal factor levels due to the random nature of X inactivation, where one of the two X chromosomes is silenced in each cell. If, by chance, a larger proportion of cells inactivate the normal X chromosome, the carrier female may exhibit mild bleeding symptoms. Biochemistry of hemophilia. Understanding hemophilia requires a grasp of the biochemical roles of coagulation factors 8 and 9 within the intricate cascade of blood clot formation, known as hemostasis. Factor 8, F8, the protein deficient in hemophilia A, is a large glycoprotein that serves as a crucial cofactor in the intrinsic pathway of coagulation. The F8 gene, located at XQ28, provides the blueprint for this protein. F8 circulates in the bloodstream bound to von Willebrand factor, VWF, which stabilizes it and protects it from premature degradation. Upon activation by trace amounts of thrombin, factor 2A, F8 is converted to its active form, factor 8A, F8A. F8A then binds to the surface of activated platelets and endothelial cells, forming a complex with another activated serine protease, factor 9A, F9A. This complex, known as the tenase complex, is pivotal in the coagulation cascade as it dramatically increases the efficiency of factor 10, F10 activation to factor 10A, F10A. Factor 10A is a key enzyme that initiates the common pathway of coagulation, ultimately leading to the formation of a fibrin clot. In individuals with hemophilia A, a deficiency or dysfunction of F8 disrupts the formation and function of the tenase complex. This results in a significantly reduced rate of factor 10 activation, leading to impaired thrombin generation and consequently a compromised ability to form a stable fibrin clot and stop bleeding effectively. The severity of hemophilia A directly correlates with the residual functional F8 activity. Factor 9, F9, the protein deficient in hemophilia B, is a vitamin K-dependent serine protease encoded by the F9 gene located at XQ27.1, Q27.2. Vitamin K is essential for the post-translational modification, specifically gamma carboxylation of glutamic acid residues, which is critical for its calcium binding and interaction with phospholipid surfaces, necessary for its activation and function. F9 is activated to its active form, factor 9A, F9A, through two main pathways. By factor 11A, another serine protease in the intrinsic pathway, activated by contact activation, factor 12, high molecular weight kininogen and prekallikrein. By the tissue factor, factor 7A complex in the extrinsic pathway, which is initiated upon vascular injury and exposure of TF. Once activated, F9A, in conjunction with its cofactor F8A on the platelet and endothelial cell surfaces, forms the tenase complex, as mentioned earlier. This complex is crucial for the amplification of the coagulation cascade through efficient activation of factor 10. In hemophilia B, a deficiency or dysfunction of F9 impairs the formation of the functional tenase complex. Even though the extrinsic pathway can initiate some factor 10 activation, the significant amplification provided by the F9A F8A complex is missing, leading to insufficient thrombin generation and impaired hemostasis. The severity of hemophilia B is also directly linked to the level of functional F9 activity. Cellular molecular pathways of hemophilia. The coagulation cascade, in which factors 8 and 9 play pivotal roles, is not a process that occurs in isolation within the bloodstream. It is a tightly regulated series of enzymatic reactions that are localized and modulated by interactions with various cellular components, primarily platelets and endothelial cells. Understanding these cellular interactions is crucial to fully grasp the pathophysiology of hemophilia. Platelets. These small, enucleate blood cells are essential for primary hemostasis, the initial formation of a platelet plug at the site of injury, and also provide a crucial surface for the assembly and activation of coagulation factor complexes, including the tenase complex. Adhesion and activation. Upon vascular injury, platelets adhere to the exposed subendothelial matrix, primarily collagen, via specific receptors like glycoprotein 195, GPIN5, binding to von Willebrand factor. This adhesion triggers platelet activation, leading to a change in their shape, the release of granules containing various procoagulant factors like ADP and thromboxane A2, and the expression of negatively charged phospholipids on their surface. Surface for coagulation complexes. The activated platelet surface, rich in phosphatidylserine, provides a binding site for calcium ions and the assembly of key coagulation complexes, including the tenase complex and the prothrombinase complex. This localization significantly enhances the efficiency of these enzymatic reactions, increasing the local concentration of enzymes and substrates. In hemophilia A or B, the deficiency of F8 or F9, respectively, impairs the formation of a functional tenase complex on this crucial platelet surface, hindering the downstream activation of factor 10. Endothelial cells. These cells lining the blood vessels play a complex and dynamic role in hemostasis, exhibiting both procoagulant and anticoagulant properties. Procoagulant functions. Upon injury, endothelial cells can express tissue factor, TF, initiating the extrinsic pathway of coagulation. They also release von Willebrand factor, which is essential for platelet adhesion and stabilizes factor 8. The surface of activated or damaged endothelial cells can also support the assembly of coagulation complexes, although generally to a lesser extent than platelets. Anticoagulant functions. Under normal, uninjured conditions, endothelial cells express various anticoagulant molecules, such as thrombomodulin, which activates protein C, a natural anticoagulant, heparan sulfate, which enhances the activity of antithrombin, and tissue factor pathway inhibitor, TFPI. A disruption in the balance between the procoagulant and anticoagulant functions of endothelial cells can influence the bleeding phenotype in hemophilia, although the primary defect lies in the clotting factors themselves. Cellular signaling pathways. The activation of platelets and endothelial cells involves intricate intracellular signaling pathways that are triggered by various stimuli, e.g., collagen, thrombin, ADP. These pathways lead to changes in cell morphology, receptor expression, and the release of signaling molecules that further amplify the hemostatic response. While the primary defect in hemophilia is the lack of functional F8, the downstream consequences impact these cellular interactions and signaling events, leading to a failure to generate sufficient thrombin to stabilize the platelet plug and form a robust fibrin clot. Furthermore, the interaction between the coagulation system and the cellular response to injury and inflammation is increasingly recognized. For example, thrombin itself is a potent signaling molecule that can activate various cell types and influence inflammatory processes. The dysregulation of thrombin generation in hemophilia can have broader implications beyond just the immediate bleeding risk. In summary, the coagulation cascade is not a fluid phase reaction, but rather a tightly orchestrated process that relies heavily on the surfaces and signaling molecules of platelets and endothelial cells. The deficiencies in factors 8 or 9 in hemophilia disrupt the formation of key enzyme complexes on these cellular surfaces, leading to a failure to generate sufficient thrombin and achieve effective hemostasis. Pathogenetics of hemophilia. Pathogenetics refers to the mechanisms by which genetic mutations lead to the development and manifestation of disease. In the context of hemophilia A and B, the diverse mutations in the F8 and F9 genes ultimately converge on a single outcome, a deficiency in the functional activity of the respective clotting factor. However, the specific type and location of the mutation significantly influence the severity of the hemophilia and can also impact the risk of developing inhibitors. Impact of mutation type on severity: null. Mutations that lead to a complete absence of the F8 or F9 protein, such as large deletions, nonsense mutations early in the gene, or frameshift mutations, typically result in severe hemophilia, factor activity less than 1%. Missense mutations. Single amino acid substitutions can have variable effects. Some missense mutations may disrupt critical functional domains of the protein, leading to severe hemophilia, while others may result in a partially functional protein, causing moderate 1 to 5% activity or mild 5–40% activity hemophilia. Splice site mutations. These mutations affect the processing of pre-mRNA, potentially leading to the production of non-functional or partially functional protein, resulting in a spectrum of disease severity. Inversions. In F8, the common intron 22 inversion in the F8 gene typically results in severe hemophilia A due to the disruption of the F3 protein. Promoter mutations. Mutations in the promoter region of the F8 or F9 genes can affect the level of gene transcription, leading to reduced protein production and milder forms of hemophilia. The correlation between genotype, the specific genetic mutation, and phenotype, the observed clinical severity, in hemophilia is often complex and not always perfectly predictable. While certain types of mutations are strongly associated with severe disease, there can be variability even within individuals with the same mutation due to other genetic and environmental factors. Inhibitor development. A significant complication in the pathogenetics of hemophilia is the development of inhibitors, which are antibodies, typically IgG, that the patient's immune system produces against the infused clotting factor. Inhibitor development renders standard replacement therapy less effective or completely ineffective. Genetic predisposition. Certain genetic factors, including the specific F8 or F9 gene mutation, as well as variations in genes related to the immune system, e.g., MHC class II genes, are thought to increase the risk of inhibitor development in hemophilia A. For example, individuals with severe null mutations in the F8 gene have a higher incidence of inhibitor development compared to those with mutations allowing for some endogenous F3 production. The association between specific F9 mutations and inhibitor development in hemophilia B is less well defined, but still exists. Immune response. The introduction of exogenous clotting factor in individuals with a completely absent or severely altered endogenous protein can be recognized by the immune system as foreign, triggering an immune response and the production of inhibitory antibodies. Clinical factors. Factors such as the intensity and type of factor replacement therapy, as well as the patient's age at first exposure to factor concentrate, may also play a role in inhibitor development. The presence of inhibitors significantly complicates the management of hemophilia, requiring the use of bypassing agents or immune tolerance induction, ITI therapy, to try to eliminate the inhibitors. In summary, The pathogenetics of hemophilia involves understanding how a wide array of mutations in the F8 and F9 genes lead to deficient clotting factor activity, and how these genetic defects, along with immune system factors, can result in the challenging complication of inhibitor development. Screening and techniques for hemophilia. The accurate diagnosis of hemophilia and the identification of carriers rely on a combination of clinical assessment, family history, and sophisticated laboratory techniques. Activated partial thromboplastin time, APTT. This is a common screening test that measures the integrity of the intrinsic and common pathways of coagulation. In individuals with hemophilia A or B, the APTT is typically prolonged due to the deficiency of factors VIII or INCs, respectively. However, a normal APTT does not entirely rule out mild hemophilia. Prothrombin time, PT. This test assesses the extrinsic and common pathways of coagulation and is usually normal in individuals with hemophilia A or B, as factors VIII and INCs are primarily involved in the intrinsic pathway. Platelet count and function tests. These are typically normal in individuals with hemophilia, as the primary defect lies in the coagulation factors. Diagnostic assays, factor VIII and factor IX activity assays. These are quantitative tests that directly measure the functional activity levels of F8 and F9 in the patient's plasma. The results are usually expressed as a percentage of the normal mean. These assays are crucial for confirming the diagnosis of hemophilia, differentiating between hemophilia A and B, and determining the severity of the condition. Genetic testing. Genetic analysis plays an increasingly vital role in the diagnosis and management of hemophilia. It can confirm the diagnosis, identify the specific underlying mutation, facilitate carrier detection in females, and enable prenatal diagnosis. Various techniques are employed. Targeted mutation analysis. If a specific mutation is already known within a family, this technique can quickly and efficiently screen other family members for that particular mutation. Sanger sequencing. This traditional method allows for the direct sequencing of specific exons or the entire coding region of the F8 or F9 gene to identify a wide range of mutations, including point mutations, small insertions, and deletions. Next-generation sequencing, NGS. NGS technologies allow for the simultaneous sequencing of multiple genes or even the entire genome, significantly increasing the speed and efficiency of mutation detection, especially when the specific mutation is unknown or when screening for multiple potential genetic factors. Multiplex ligation-dependent probe amplification, MLPA. This technique is used to detect larger genomic rearrangements, such as deletions or duplications of one or more exons within the F8 or F9 genes, which might not be easily identified by standard sequencing methods. The MLPA technique uses probes that hybridize to specific DNA sequences within the target gene. The amount of PCR product generated for each probe is proportional to the copy number of that sequence. Chromosomal microarray analysis, CMA. While not routinely used for point mutation detection in hemophilia, CMA can identify larger chromosomal abnormalities, such as deletions or duplications involving the X chromosome, which could, in rare cases, lead to hemophilia in females, e.g. in Turner syndrome with a mutated X. RNA analysis. In some cases, particularly when splice site mutations are suspected, RNA analysis can be performed to assess the impact of the mutation on mRNA processing and identify aberrant transcripts. Carrier detection. Identifying carrier females is crucial for genetic counseling and family planning. Mutation-specific testing. If the specific mutation in the F8 or F9 gene has been identified in an affected male in the family, carrier testing for female relatives is highly accurate. Factor VIII or IN activity assays. While factor activity levels can be lower in some carriers due to skewed exon activation, the overlap with normal female ranges can make this method less reliable for definitive carrier status, especially in the mild range. Genetic testing for the specific mutation is the preferred method when available. Linkage analysis. In families where the mutation is unknown or difficult to identify, linkage analysis using polymorphic DNA markers closely linked to the F8 or F9 genes can be used to track the inheritance of the chromosome region containing the hemophilia gene. However, with the advancements in direct gene sequencing, this method is less frequently used. Prenatal diagnosis. For families with a known risk of hemophilia, prenatal diagnosis options are available. Chorionic villus sampling, CVS. performed typically between 10 and 13 weeks of gestation. CVS involves obtaining a small sample of placental tissue containing fetal cells for genetic analysis. Amniocentesis, usually performed between 15 and 20 weeks of gestation. Amniocentesis involves extracting a small amount of amniotic fluid containing fetal cells for genetic analysis. Pre-implantation genetic diagnosis (PGD) for couples undergoing in vitro fertilization (IVF). PGD allows for the genetic testing of embryos before implantation, enabling the selection of unaffected embryos for transfer. The advancements in these screening and diagnostic techniques have significantly improved the ability to diagnose hemophilia early, identify carriers accurately, and provide comprehensive genetic counseling to affected families. Treatments for hemophilia. The management of hemophilia has undergone a significant transformation, evolving from primarily on-demand treatment of bleeding episodes to proactive prophylaxis and the emergence of innovative therapies aimed at long-term control and even potential cures. Replacement therapy, on-demand treatment. Historically, the primary approach was to treat bleeding episodes as they occurred with infusions of the deficient clotting factor concentrate. For hemophilia A, this involves factor VIII concentrates, and for hemophilia B, factor IX concentrates. These can be plasma-derived obtained from human plasma or recombinant, produced in laboratory settings. Prophylactic treatment. The cornerstone of modern management for moderate to severe hemophilia is regular, scheduled infusions of factor concentrate to maintain a baseline level of clotting factor activity and prevent spontaneous bleeding episodes. Prophylaxis has significantly reduced the frequency of bleeds, the development of chronic joint damage, hemophilic arthropathy, and improved the overall quality of life for individuals with hemophilia. Extended half-life factor concentrates. Recent advancements have led to the development of F8 and F9 concentrates with prolonged half-lives in the circulation. These modified proteins require less frequent infusions, reducing the treatment burden and improving adherence to prophylactic regimens. Various methods are used to extend half-life, including echylation, attaching polyethylene glycol molecules, and fusion with other proteins like albumin or the Fc portion of immunoglobulin G, IgG. Treatment of inhibitors. A significant complication is the development of inhibitors, antibodies against the infused clotting factor. Management strategies for patients with inhibitors include bypassing agents. These medications, such as recombinant activated factor VII and activated prothrombin complex concentrates, APCCs, promote blood clotting through alternative pathways. Emicizumab. This is a bispecific antibody approved for hemophilia A with and without inhibitors. It mimics the cofactor function of F3 by bridging factor IXA and factor X, effectively promoting blood clot formation. Emicizumab is administered via subcutaneous injection, offering a less frequent and often more effective prophylactic option, particularly for patients with inhibitors. Immune tolerance induction (ITI). This therapy aims to eliminate the inhibitor by regular, frequent infusions of high doses of the deficient clotting factor. ITI can be successful in a significant proportion of patients, allowing them to return to standard replacement therapy. Gene therapy. Gene therapy represents a revolutionary approach with the potential to provide long-term correction of the underlying genetic defect. Adeno-associated virus, AAV vectors. The most common approach involves using AAV vectors to deliver a functional copy of the F8 or F9 gene to liver cells, the primary site of clotting factor production. Several AAV-based gene therapies for hemophilia A and B have shown promising results in clinical trials, leading to sustained increases in clotting factor levels and a significant reduction in bleeding rates and the need for prophylactic infusions. Some gene therapies have received regulatory approval. While gene therapy offers great promise, challenges remain, including the long-term durability of the treatment effect, potential immune responses to the AAV vector or the transgene product, the variability in patient responses, and the cost and accessibility of these therapies. Ongoing research focuses on optimizing vector design, improving safety and efficacy, and exploring alternative gene delivery methods. Emerging therapies. Beyond gene therapy, other novel therapeutic strategies are under development. Fitusiran, a small interfering RNA, siRNA, that targets antithrombin, a natural anticoagulant, By reducing antithrombin levels, fitusiran aims to rebalance hemostasis and promote thrombin generation in individuals with hemophilia A or B, regardless of inhibitor status. It is administered via subcutaneous injection. Concizumab, a humanized monoclonal antibody that blocks tissue factor pathway inhibitor, TFPI, another natural anticoagulant. By inhibiting TFPI, concizumab aims to enhance thrombin generation in hemophilia A and B. It is also administered subcutaneously. CRISPR-Cas9 gene editing. This technology holds the potential for in vivo correction of the mutated F8 or F9 gene. While still in earlier stages of clinical development for hemophilia, CRISPR-based therapies could offer a permanent and potentially curative treatment. Comprehensive care, optimal management of hemophilia requires a multidisciplinary approach involving hematologists, nurses, physiotherapists, orthopedic surgeons, dentists, and social workers. Comprehensive care includes education and self-management training, empowering patients and their families to understand the condition, recognize and treat bleeds early, and administer factor concentrates at home. Musculoskeletal management, preventing and treating joint and muscle bleeds through physiotherapy, exercise programs to strengthen supporting muscles, and, in some cases, orthopedic interventions. Pain management, addressing acute and chronic pain associated with bleeding episodes and hemophilic arthropathy. Psychosocial support, providing emotional and social support to patients and their families to cope with the challenges of living with a chronic bleeding disorder. Dental procedures in individuals with hemophilia require careful planning and coordination with the hematology team to minimize the risk of bleeding. This may involve pre-treatment factor infusions and the use of local hemostatic measures. The treatment landscape for hemophilia is rapidly evolving, offering increasing options for effective bleed prevention, management of complications, and the exciting prospect of long-term therapies and potential cures. This brings us to the conclusion of our exploration into the royal disease, hemophilia. We've journeyed through its historical impact on European monarchies, delved into the intricacies of its molecular and medical genetics, and examined the biochemical pathways and pathogenetics that lead to its clinical manifestations. We've also explored the screening techniques used for diagnosis and the remarkable advancements in treatment, including the revolutionary potential of gene therapy. The story of hemophilia serves as a powerful reminder of the intersection between genetics and history, and how a single gene mutation can have profound and far-reaching consequences, shaping not only individual lives, but also the course of nations. The ongoing scientific advancements offer hope for a future where the challenges of hemophilia are significantly mitigated. Thank you for joining me on this episode of Decoding Dynasty. If you enjoyed this exploration of hemophilia, please subscribe to my channel and stay tuned for next episodes of Decoding Dynasty for more deep dives into the genetics of historical figures and events. You can also follow me on my Telegram channel for updates and articles of this video. Until next time, this is Zara, your geneticist host, signing off.